Thank you very much for coming to our symposium. We are glad to show you our research content and also hope to have an in-depth academic exchange with you. I'm the chair of this symposium, Lin Sun, Associate Professor of Weifang Medical University. I graduated from Shandong University, studied for 1 year under David Yao at the Chinese University of Hong Kong, and I'm currently conducting research on post-traumatic stress disorder, PTSD, in Weifang, Shandong, China, the world capital of kite flying and the international city of peace. The research direction of our group is related to the treatment and the pathological mechanism of PTSD. Since the team was established, we have carried out multi-symptom and multi-dimensional experimental research around PTSD and other psychiatric disorders. Moreover, the research was supported by the National Natural Science Foundation of China, NSFC, the Hong Kong Narcotics Division, the Chinese Academy of Science Innovation Program Pilot Project, the NSFC Outstanding Youth Fund, and so on. Besides, our team members have published 12 SCI papers as the first author or corresponding authors about PTSD. The topic of our symposium is ketamine and ketamine metabolite, pharmacology, therapeutic mechanism in mental disorder. Next, a member of our team will introduce the current research progress we found to you. Their themes are HCN1 ion channel in the hippocampal C1 region was involved in ketamine-induced antidepressant action and increased BDNF levels in mice.
Hello, ladies and gentlemen. It is a pleasure to be here with you today. First, I'd like to introduce myself. I'm Xu Yanling, a master degree candidate from Qingdao University. Today, I'm going to talk about the HCN1 ion channel in the hippocampal C1 region was involved in ketamine-induced antidepressant action and increased BDNF levels in mice. In the first part, I will introduce the background of our study. Then, in the second part, I will show you the experiment design. In the third part, I will show you the study results. Finally, I'll go on to talk about the conclusion of this research. As we all know, major depressive disorder is a prevalent neuropsychiatric disease that carries a staggering global burden. A meta-analysis study found that there were 6 to 9% people have this disorder in China. This therapeutic lag highlights the need for new antidepressants that provide rapid relief of depressive symptoms. Indeed, depression pathology possibly arises due to changes in excitability in the limbic circuitry. Ion channels, in particular HCN channels, are key regulators of neuronal excitability. As we all know, neuronal signaling within a region and at a circuit level is affected by intrinsic excitability and the distribution of ion channels. Dysfunction of ion channels can lead to abnormal excitability and subsequent neuronal dysfunction. HCN channels are regulating hyperpolarization activated currents and affecting neuronal excitability. Although there are 4 isoforms of HCN channels, HCN1 is the predominant isoform expressed in the hippocampus, and the other like neural cortex and the cerebellar cortex. HCN channels are highly expressed in pyramidal neurons in the hippocampus C1 region. The HCN1 channel in CA1 has essential roles in the modulation of neuronal excitability under physiological conditions. Moreover, it plays a significant and complex role in neuropsychiatric diseases. Moreover, some anesthetic doses of ketamine produce rapid and sustained antidepressant responses in patients suffering from major depressive disorder. The search for pathways of ketamine's action has identified potential targets of HCN1 channel, suggesting exciting new drug discovery targets. Some reports indicate that ketamine caused direct neurophysiological effects on pyramidal neurons of hippocampus CA1 region. BDNF is a critical signaling molecule for nervous system development that continues to play an important role in the maintenance and survival of neurons. Acute administration of ketamine increases the expression of BDNF protein, which induces antidepressant effects. mTOR has well-known functions in protein synthesis, synapse formation, and neurogenesis in neurons. Therefore, the research objectives are as follows. First, we applied 8-BRCAMP and ZD7288, an agonist and inhibitor for HCN1 ion channel, into the hippocampus CA1 region and investigate its effect on rapid antidepressant effect of ketamine. Then, we examine whether BDNF and mTOR is involved in the regulation of ketamine antidepressant effect through HCN1 channel. Now, let's move to the methods. And first, 8-BRCAMP or ZD7288 were isolated in order to regulate the HCN1 channel in the hippocampus CA1 region. After 7 days recovery, the forced swimming test was used to evaluate the antidepressant-like effect of drugs and investigate their action mechanisms. To minimize variability in baseline immobility between animals, each mouse was pre-exposed to forced swimming for 15 minutes, 24 hours before the test session. Finally, the mice were sacrificed after 20 minutes of forced swimming test. Now that we have seen the results parts, ketamine significantly decreased the passive activity time of mice. High dose of 8-BRCAMP was identified to be optimal in blocking the actions of ketamine. Ketamine significantly increased the latency to immobility. However, the high dose of 8-BRCAMP didn't impact the ketamine-induced increase in the latency to immobility. We didn't find statistically significant or main effect of pretreatment with high dose of 8-BRCAMP, while there was a significant interaction between pretreatment with high dose of 8-BRCAMP and ketamine, indicating that the high dose of 8-BRCAMP decreased the latency to immobility, which was induced by ketamine. Mice pretreatment with 8-BRCAMP and ketamine started variations in the expression of BDNF, mTOR, and pMTOR in the hippocampus CA1. The results of the Western blotting showed that ketamine increased the expression of BDNF and the phosphorylation of mTOR. The level of BDNF in the group 8-BRCAMP high dose ketamine was significantly lower than that in the group vehicle ketamine. While 8-BRCAMP cannot play its role alone, The ketamine-induced increase in phosphorylation of mTOR was not affected by pretreatment with 8-BRCAM. In addition, all treatments had no effects on the expression of mTOR. Now let's look at the effects of pretreatment with ZD-7288 on the action of ketamine. As is shown in this figure, pretreatment with ZD-7288 and treatment with ketamine didn't have a significant interaction effect. The current study results showed that ketamine increased the protein expression of BDNF in hippocampus A1. Pretreatment with ZD-7288 and treatment with ketamine could enhance this effect, but only pretreatment with ZD-7288, but not treatment with ketamine couldn't improve the protein expression of BDNF in hippocampus A1. In addition, all treatments had no effects on the expression of mTOR and pMTOR. So, our study conclusions are as follows. We found that pretreatment with HCN1 ion channel antagonist 8-BRCAM attenuated the ketamine-induced decreasing of immobility time in phosphosphamine test. And pretreatment with 8-BRCAM attenuated ketamine-induced increasing of expression of BDNF, while ZD-7288 potentiated this effect. And we also found that pretreatment with 8-BRCAM or ZD-7288 had no effect on ketamine-induced increases in mTOR phosphorylation. In brief, there are several advantages and disadvantages in our study. That's all I want to say for now on. Thank you.
One-time treatment of ketamine alleviates fair memory and spatial combination deficits in PTSD-like models, where BDNF signaling and post-synaptic density magnified of hippocampus and amygdala.
I'm a member of the myself and a graduate student of Pharmacology University, and I'm currently conducting research on post-traumatic stress disorder in Weifang, Shandong, China. The topic of my report is one-time treatment of ketamine elevates fair memory and specific cognition deficits in PTSD-like models via BDNF signaling and post-synaptic density 95 of hippocampus and amygdala. Now, I'm going to make my presentation. I will present my report in terms of background, method, result, conclusion, and prospect. Part 1, background. And I will start by telling you a little bit about the relationship between PTSD and ketamine. Post-traumatic stress disorder is characterized by exaggerated response to traumatic experience and impaired fear extinction, and may accompany cognitive impairments and learning deficits. Ketamine can rapidly and significantly reduce the severity of fear symptoms in PTSD patients. Its pharmacological mechanism has yet to be determined with the reading incidence of traumatic events, such as natural disasters, traffic accidents, violent conflicts. The incidence of PTSD is also increasing. To be specific, when people who have experienced traffic events are re-exposed to traumatic-related situations, the inappropriate fear response may occur later. John Watson, an American behaviorist psychologist, once conducted an interesting experiment as shown in the figure. When natural stimuli and versus stimuli are repeatedly presented to infants, even when natural stimuli alone are present to infants after a period of time, they will trigger inappropriate fear. Memory impairment is considered part of the diagnostic criteria for PTSD. PTSD damages spatial memory in rodents by disturbing several aspects of hippocampal function. Given the important role of the hippocampus in spatial memory in rodents, it is reasonable to suggest that the PTSD in rodents can cause spatial memory difficulties. In addition, the acquisition and retrieval of contextual file memory requires coordinated neural activity present as encoded information in hippocampus and conveying to the medial peripheral left and amygdala for contextual fair condition. Therefore, based on ketamine reduced neural activity in the bilateral amygdala hippocampal complex, we reason that the action of ketamine in HIV and AMG could also influence the fair extinction. And then we mount to molecular neurobiology. Post-synaptic density protein 95 is a synaptic protein, regulating glutamate receptor acquiring synaptic stability and certain types of memory. Brain-derived neurotraffic factor is a neurotransferrin that was identified as the critical factor that mediates synaptic plasticity associated with learning and memory. An existing study suggests that BDNF signaling promotes PS95 expression in synapses and dendritic spans, regulates the postsynaptic localization of PS95 via the type B signaling pathways. Thus, the present study emphasized that ketamine could increase the BDNF protein levels in the HIP and AMG, thereby upregulating BDNF signaling, transduction, and activating expression of PS95, which was possible associated to the improvement of PTSD symptoms. Part 2, method. In this study, rats were divided into control group and the PTSD group, and the PTSD group was divided into staging groups 5, 10, 15, 20 mg killer according to ketamine concentration. This is the flowchart of the whole experiment. The rats were modeled for PTSD after 7 days of acclimation, and the fear memory was tested by contextual fear paradigm, and the spatial memory was tested by mirror watermice after a 40 days clam period. And this is our experiment equipment model. At the level of molecular biology, we will detect the plasma cholesterol content of rats in each group and detect the expression of BDNF and PS95 at the protein and gene levels. Next comes the presentation of results. The first is the detection effect of SPS&S procedure for PTSD modeling. And here we see that the PTSD group have difficulty in fear memory and spatial cognition. the expressions of BDNF and PSD-95 in hippocampus and amygdala of PTSD group were lower than those of controls. And the MIA expression of BDNF and PSD-95 in hippocampus and amygdala decreased in PTSD group. After the rats were intraperitoneally injected with ketamine according to the concentration granted, these graphs show that PTSD ketamine 15 group can significantly reduce the time of freezing, improve the spatial memory, and increase the expression of BDNF and PSD-95 protein in hippocampus and amygdala of PTSD rats. And the rates of BDNF and PS95 positive cells and the expression of MIA in hippocampus and amygdala was significantly increased in PTSD ketamine 15 group. According to the above result, ketamine reduced significantly the freezing time 24 hours after administration, and ketamine with dose of 15 mg/kg could reduce significantly the time of regressions and improve the number of crossings in the MWM and 48 hours after administration, which could be interpreted as a remission of fear and spatial memory deficits of PTSD symptoms in these ketamine-treated rats. This finding disagreed with previous reports that PTSD associated with multiple force shocks did not affect spatial memory, owing to the fear memory caused by the multiple force shocks is divided with spatial memory in the HIP. This may be due to division of neurons in different hippocampal subregions. And the ketamine induced significantly impression changes of glutamate reports in brain area relevant for renal and contextual fear memory. This may explain the result that ketamine can significantly reduce the freezing time as well as improve spatial memory and learning ability of SPS&S rats. In addition, it is noteworthy that there was no significantly response to the dose of 5 and 20 mg/kg ketamine. However, ketamine could provide prosthetic effects include negative cognition symptoms and deficits with the dose of higher than 20 mg/kg. It might be the compensatory consequence of an increasing glutamate release induced by higher dose ketamine treatment given to rats 24 hours area, and higher dose of ketamine in the brain had a negative effect in potentiating the oxidative stress. Further study revealed that ketamine could improve the number of BDNF positive cells in the HIP and MG. The decreased level of BDNF MIA and protein were reversed by ketamine after PTSD training, which was consistent with the previous study showing ketamine increases pro-BDNF expression after single dose. This result indicated that ketamine might prevent the degradation of protein or enhance the expression of protein where the static plasmic MIA pool and involved in the extinction of fear memory. Therefore, PST95 itself and its in action with BDNF signaling might be implicated in the retrieval of frame memory, and the lower PTSD-like expression might be the key factor in spatial cognitive dysfunction, which might depend on rapid activation of the mammalian target of BDNF protein in the encephalic region. Together, these findings support the hypothesis that ketamine enhancement of extinction resulted from increased synaptic function in the HIP and AMG. Previous studies have suggested that endocytosis of AMP reporter was necessary for memory remodeling after retrieval, while AMD reporter activation was controlled by AMP reporter endocytosis during hippocampus LTD. When post-95 is recruited into synapse, AMTA reporters may migrate and aggregate, thus affecting the morphologic function, plasticity of synapse, and endocytosis of AMP reporters. Therefore, this research presented that one-time treatment of ketamine could regulate the localization of signaling by AMD reporters and elevated frame memory and spatial cognition of PTSD symptoms by upregulating BDNF and BDNF-related PSD95 signaling, and it might reverse the loss of synaptic connectivity between the HIP and the AMG, which caused by traumatic stress. From what has been discussed above, we came to the conclusion. Our study proves that ketamine enhances the expression of extinction via BDNF signaling and neuronal circuits between HIP and AMG. The activation of BDNF pathway participate in therapeutic efforts for BDNF and pathway prospects. In this study, the effects of ketamine on frame memory and spatial memory were confirmed, but the effects of ketamine on specific time window of frame memory were not involved. Ketamine is also widely limited in anti-PTSD treatment because it's potentially toxic and addictive properties. Therefore, we will focus on the key ketamine metabolite 2-6-HNK and explore its pharmacological effects in specific time windows of frame memory. That's all for my report. Thank you for your attention.
Ketamine is the focus of our research. The following members will tell you about the different roles of ketamine in PTSD. Their themes are: ketamine impairs cognitive ability in rats. Evidence from ASST.
Okay, let's turn to the study of the other effects of ketamine. Maybe the good news of the first two studies has swept away the gloom of your life, but my findings, the side effects of ketamine on cognitive impairment may play a bad role. Put a little haze on your good mood and consider it a little sacrifice for science. Well, let's take a look at this abominable title. Ketamine impairs cognitive ability in rats. Evidence from ASST. I'm the reporter of this study, Lydia. The research director of our research group is post-traumatic stress disorder. Lecturers have already explained the relevant knowledge and the definition of PTSD. I would like to express my view. After being subjected to intense stress that is unforeseeable and life-threatening, the patient's brain will get sick, just like our fever after cold stimulation will show a variety of symptoms. Because PTSD itself is a comprehensive mental disorder with multi-types, multi-dimensional characteristics, and so we have done targeted research on its different symptoms. Some recent studies have found that people with PTSD will have severe cognitive impairment, while persistent cognitive impairment will maintain and develop other symptoms of PTSD, which has attracted our attention. For this reason, I designed the following experiment. 8-week-old rats were divided into 4 groups with 10 rats in each group. The chart is as follows. The rats were normally raised in the laboratory for 1 week after being purchased from the Pharmacology University to maintain the normal development of cognitive function of rats. A week later, the rats were subjected to the classic PTSD modeling operation called SPS&S. 2 weeks later, the model was tested and ketamine was injected intraperitoneally. Finally, the cognitive ability of the rats was tested by attention site transfer test and one maze. And the specific experimental design is as follows. The model is detected using open field and elevated cross maze paradigm. Single injection dose of ketamine is 15 mg per kilogram. This is the most important experimental paradigm of this study, attention site transfer task, which simulates the human Wisconsin card sorting test and is an important test for testing cognitive flexibility in rats, because the executive function detected at the same time in this experimental belongs to the core of cognitive function. This test fits the purpose of this study very well. The picture on the left is the top view of the experimental device. The experiment is divided into 3 parts: publication, training, and testing. The rats had been restricted for 3 days before the publication stage. When they were hungry and hungry for food in the experiment, the subject opened the front and the rat had 2 options. 1 container on each side can be dug, but only 1 container contained food, and the containers were filled with padding enough to bury the reward. Rats only need to find the right food 3 times in a limited time to pass. During the training phase, the rats were required to simply identify a link, and the reward could be found. According to the scented simulation hint, it was thought that the right thing could be done 6 times in a row, and each container was still filled with chlorine. In the testing stage, the caution material is replaced by the digging medium, and the role of the digging medium is different in the 6 periods of this stage. The table on the right shows the detailed settings for the retaining SD freeze and the 6 test freezes. It's worth noting that the ED freeze will replace cues with media, and the smell will become irrelevant cues, which is called out-of-dimensional conversion, and rats need to relearn the matching of rewards and cues, which is the most difficult part of the experimental process. If you are a little confused about this paradigm, but don't worry, it's not your problem, because this experiment is designed for rats, and we have a Wisconsin card sorting test, I think you can try to test it so you can sleep at ease. So let's review the experimental results. The statistics of the results are done by another college. The above four color pictures are the tracks of the elevated cross maze and the open field. And the first two pictures are the tracks of the elevated cross maze of the rats in the control group and the model group, respectively. The following bar chart shows the following results. The number of times of entering the open arm and the time of staying in the open arm in the control group were significantly more than those in the model group. In addition, there was a significant difference in the distance between the two groups in the central area of the open field. Two behavioral studies show that the authority level of rats treated with SPS&S is significantly increased. Of course, it also shows that our modeling process is effective. Let's take a look at the cognitive ability of the rats. That is, the ASIT experiment just now. I'm not sure if the rats will feel tired in this experiment, but what is certain is that my college must be exhausted because the experiment procedure are very tedious. Not to mention is that he has to watch the rats eat hungrily. Let's cheer up and listen to me talk about the results of the experiment. Finger A is a three-dimensional diagram of the experimental device, and finger B is a statistical chart of the number of experiments. We can see that the number of experiments in rats treated with SPS&S and after a single dose of ketamine is significantly more than that in the corresponding control group. This result is reflected in both intradimensional and out-of-dimensional changes. This shows that traumatic stress causes some damage to the cognitive flexibility of rats, and ketamine is also potentially harmful to the cognitive ability of rats. C and D are error rate and delay time respectively, both of which reflect the cognitive impairment of rats caused by SPS&S and ketamine in different degrees. In addition, we also did another experiment to test the cognitive ability of rats, the one-maze experiment, which reflected the working memory ability of rats by measuring the automatic alternation rate and the number of times they entered the arm. As a classic paradigm for measuring cognitive ability, this experiment also reflects the same results as ASST. Point. The progress of my experiment is over. I believe you have a little more doubt about ketamine, a drug full of hope. As Holmes, who discovered this doubt, we will also unveil the view of ketamine and spare no effort to actively explore in the field of cognitive impairment in PTSD. If you are interested in this research or would like to communicate with me, please contact this email address. My colleague will next introduce to you a new drug to cure PTSD, HNK, a derivative of ketamine. I believe her story will give you new hope.
2R6R hydroxyketamine with gluon A1 mediated synaptic plasticity alleviate the PTSD-like effects in the prefrontal cortex.
Through the previous presentation, we know that post-traumatic stress disorder (PTSD) is a debilitating mental disorder with high mobility, which has a great hazard of social and economic relevance. Previous studies by the group have shown that ketamine is able to alleviate PTSD effects in the rat model, included by single prolonged stress and electric foot shock (SPS&S), but its potential for widespread clinical use is limited owing to its abuse liability and capacity to produce dissociative effects. In the following, I will talk about the effect of 2R6R18K on PTSD in our team research. The title of this speech is 2R6R18K will GNU AE mediated synaptical plasticity elevated the PTSD-like effect in the prefrontal context. I am the reporter of the study, Yalin Du, Department of Clinical Medicine, Weifang Medicine University. The whole presentation is divided into 4 parts, which are introduction, missions, results, and conclusion. The first part is introduction. PTSD is a developmental disorder with high morbidity. People will recall some traumatic-related memories, extreme avoidance, and overvigilance of traumatic events. After experiencing or witnessing one or more major traumatic events, such as Dunn Street, which has a great heart of social and economic relevance. However, exempt pharmacotherapies or PTSD required long-term use to maintain effectiveness, and the side effect was also enormous. Demonstrating a major unmet medicine need to find more effective treatment for PTSD. For these reasons, new target and research priority need to be identified to improve the symptoms of PTSD. Let's start from that. 2R6R hydroxyketamine ketamine. 2R6R18K is the major metabolite of RS-ketamine, found in the plasma and brain in vivo after its administration to humans and other mammalian species. Preclinical studies indicate that 2R6R18K exerts at dispersion element behavior actions without a social side effect. Under normal conditions, stimulation of presynaptic neurons to release glutamate, activity post-synaptic glutamate, alpha-amino 3 structures, 5-methanol, 4-isoxysulfuric acid, AMPA reporters, result in despolarization, which enters the activation of multiple intracellular pathways. Clinical stress is known to cancer neural atrophy and reduce the number of synapses, involving the revolution of mind connection and behavior, and glutamate synapses are the main form of synapses. As reviewed, stress impairs the integrity of signaling via glutamate synapses in multiple ways, reducing the number of AMPA reporters in the synapses and displaying brain-derived neurotrophic factor BDNF release and downstream cellular pathways. Recent clinical evidence suggests that 2R6R18K modulates glutamatergic neurotransmission and downstream signaling pathway in the prefrontal context. Therefore, we propose the hypothesis that 2R6R18K activates the downstream BDNF signaling pathway by increasing postsynaptic glutamate release in prefrontal cortex, which in turn enhances synaptic plasticity and achieves the effect of improving PTSD symptoms. We decided the following experiment. A total of 16 8-week-old steroids with a weight of 220 and 215 provided by the Animal Sanctuary of Weifang Medicine University was selected for this study. The rat need to be familiar with the new environment. All experiments we did were conducted in accordance with the guidelines of National Institute of Health and were approved by the Animal Care and Use Committee of Weifang Medicine University. Among the various animals models of PTSD, the SPS&S procedure was used in our study. That a single inescapable electric foot shock was given to rats immediately after SPS&S procedures. Rats were acclimated for 15 days after SPS&S modeling and randomly divided into 6 groups. Control group, SPS&S group, SPS&S cell group, 2R6R HNK groups. The concentration of 2R6R HNK we have selected 3 types according to the concentration guidelines, which are 20, 15, and 100 UMR. 10 rats in each group. After 15 days of the SPS&S procedure, 2R6R was administered to the experiment rat, while the SPS&S cell group was given the same volume of 0.9% saline. After 24 hours of 2R6R or saline treatment, the experiment animals was acclimated in the experiment environment for 1 hour. Behavior test was performed in a quiet and noiseless environment, and each rat was wiped clean with 75% medicine alcohol after the behavior test before the next one to prevent interference with the experiment of other rats. After behavior test, significant was performed for molecular acids, which was for behavior test, open-fired test, OFT, 11th plus MET test, EPMT, frozen behavior test, FBT, and foxed swim test, FST. At the same time, Western blot and immunofluorescence (IF) was used to analyze protein, GLUAE, and BDNF expression levels, and transmission electron microscopy (TEM) was used to observe changes in synapses ultrastructure. The next part is the results. The result of this experiment will be shown below. In open-fired test, which was for experiment indicators to study the anxiety behavior of SPS&S rats separately. They are the time spent in the sensory area, distance in the sensory area, upright numbers, and the crossing numbers. In 11th plus MET test, which was 2 experiment indicators that enters in the open arms and time spent in the open arms. Firstly, the result of the SPS&S procedure are shown to you. The 4 images of the respective video tracking images of the open-fired test and the 11th plus MET test. Figures A and C are the control group, and figures B and D are the SPS&S group. Figures E and G show that the SPS&S group, the time spent and distance in sensory area are significantly lower compared with the control group, as well as the upright numbers and crossing numbers was also noticeably decreased, as shown in figures F and H. This indicated that SPS&S rats have reduced exploratory activity and significantly anxiety behaviors. Compared with the control group, the SPS&S group enters in the open arms and time spent in the open arms are significantly reduced. Figures H and I. The change in behavior activity of the SPS and S right in the open-fired test and 11th plus mass test indicated that a decrease in autonomous activity, an increase in intention, and the presence of significant anxiety behavior. Figures G show that the result of freezing behavior test, the freezing times of the SPS and S group, is significantly increased compared with the control group, and the involuntary times of the modern right increased in the focused swim test, which are obviously different compared with the control group, as shown in Figure K. This behavior data implies that there are always fear and depression-like behavior in the SPS and S group compared to the control groups. This also indicates that the SPS and S model we established was successful. The above is an analysis of the SPS and S model acid state. And then let us look at the changes in behavior and protein levels associated with the administration of different concentrations of 2R6-RHNK2 right, as well as the changes in synaptic ultrastructure. 2R6-RHNK was administered at a single dose in open-fired test. The SPS and S right showing an increase in voluntary activity and a significant difference in time spent in the central area among these groups, with evident increased time spent in the central area in SPS and S15 group compared to the SPS and SCL group. There were also variability in the upright numbers in these groups. The upright numbers with a significant increase in the SPS and S50 and SPS and S100 groups compared to the SPS and cell group, as shown in figures A and B. Figures D indicated that there is a difference in the crossing numbers in these groups, and with a significantly higher in the SPS and S100 group. Also, each phase enters in the open arms and the time spent in the open arms was different in the element plus mass test. The SPS and S15 and SPS and S100 groups had significantly increased the time spent in the open arms, and enters in the open arms also increased, but not clearly. The SPS and S100 group was able to reverse the prolongation of freezing time in the SPS and S right, and the involuntary time in the SPS and S15 and SPS and S100 groups decreased significantly, indicating that the 2R6-RHNK can improve the anxiety and depression-like behavior in the PTSD right. The following PowerPoint shows us the expression levels of the protein GLUN-AE and BDNF. From the Western blot and IF results, we were able to know that the expression levels of both GLUN-AE and BDNF was different cell elements after the administration of different concentration of 2R6-RHNK compared to the SPS and S cell group. Among them, protein expression was significantly increased in the SPS and S15 and SPS and S100 groups. The result of TEM measurement showing that the synaptic cliff-wise was reduced and synaptic interface curvature was significantly higher in the SPS and S15 group compared to the SPS and S cell group. The result of postsynaptic density, another indicator of synaptic measurement, showing that there were also increase in the SPS and S15 and SPS and S100 groups. These results suggest that 2R6-R18K is able to restore synaptic damage caused by SPS and S procedure by affecting synaptic structures in prefrontal cortex. In conclusion, we concluded that 2R6-R18K increase AMPA reporter expression and restores damage to synaptic structures caused by SPS and S procedure, alleviating of anxiety and depression-like behavior in PTSD-like, while GLUON-AE mediated synaptic plasticity. Well, this concludes my speech, and I thank you all for listening.
Thank you very much for the introduction of our team members, and we hope that our team can impress you. Despite the non-STEM academic, it hasn't hindered our relentless pursuit of science research in any way. And finally, we are honored to share our research progress and look forward to academic communication with you.